A discovery made in the early hours of Saturday at Molessi Weir has stunned scientists. During the standard May eel monitoring at 3 index sites, citizen scientists have been recording the glass eel and elver recruitment and migration as the eels congregate while attempting to find upstream passage. These work groups are part of an ongoing data assessment across the River Thames Basin District to develop mandatory eel management plans according to the European Commission regulations and the Convention on Migratory Species, CMS. During the dawn inspections, a simultaneously discovery was made of several mature yellow and silver eels with unusual glowing markings and blue gills. Closer analysis of these specimens showed that these eels were carriers of a new set of organised batch cultures of specific bacteria. While it has already been theorised that the wild eel microbiome and fish skin mucosal surfaces provide a natural niche for aquatic mucosal pathogen evolution, this weekend's discovery hints at previously unimagined possibilities. Electron micrographs of the scales and the gills of these silver eels revealed an unprecedented organisation of 3 rare strands of bacteria. The arrangement of blue cyanobacteria on the bony operculum above the gills of the eels, the only bacteria capable of oxygenic photosynthesis, suggests that a form of symbiosis previously unwitnessed has been occurring. Across the body of the eel, distinct banded markings were found of luminescent Aliivibrio fischeri, creating the glowing patterns seen in the water. The patterns marked out distinct molluscal colonies of Ideonella sakaiensis, alternating with colonies of the alkane-degrading bacterium Alcanivorax borkumensis, more commonly associated with marine environments. The presence of these bacteria as an experimental biofilm on a stock of wild silver eels has a potentially very positive outlook for the fate of these organisms. It is unusual to see free-swimming bacteria in such structured patterns, and while eels are already known for their unusual morphological change to both appearance and structure at different maturing and pigmenting stages, according to their migration across freshwater and brackish habitats, they have never been known to carry these strands of bacterial biofilm mutation. It was discovered that the eel scales magnified the effect of the photosynthetic symbionts while in sunlight near the surface of the water, but also that altered chlorophyll was responding to darker light at greater depth, particularly green light in the shadows and red light in the deeper water. Further experiments are now being undertaken to analyse bacterial strains internal to the eel gut, as well as the overall impact of these patterned eels on the unfiltered estuary waters. It is unknown what the source of this new scientific discovery is, and whether it occurs earlier in the eel's life cycle, during their complex and poorly documented movements on their mysterious migration, or before their first arrival drifted on ocean currents as leptocephali from the Sargasso Sea. However, the dissemination of these new patterned eels may well have a significant impact on the water cultures across the entire Gulf Stream, as well as the Thames tidal area in which the eels are most vulnerable, due to the survival risk in polluted waters as well as the limit on silver eel escapement routes. These new bacterial organisms may in fact be an unprecedented step to learning about dealing with several anthropogenic, oceanic and climatic factors using a new propensities of this eel biofilm for self-cleaning waters, combining self-sufficient and environmentally benign colonies, and finally taking a big step towards new forms of bioremediation of contaminated environments.